Good morning, everyone. Thank you for joining us here today. My name is Suwon Lee, and I'm a technical application specialist at IBM Technology. Our webinar today will be presented by technical application specialist Jenny Ong. She will be touching on how intravital microscopy can enable real-time visualization of antibody therapeutics action and kinetics, offering novel insights into their efficacy. If you have any questions during the webinar, do not hesitate to ask us through the Q&A box below, and we'll make sure to address them during the Q&A session. I'll now hand things over to Jenny. You may begin.
Hello and good morning, everybody. I'm Jenny Ong, Technical Application Specialist at IBM Technology. Thank you all for joining today's webinar on using intravital microscopy to evaluate the efficiency of various antibody therapeutic drugs in animal models. In this session, I will introduce how IBM Technology's intravital microscopy can be applied in the development of antibody drug therapeutics, and will also introduce several application cases in detail. And at the end of the session, I'll present a brief virtual demonstration to showcase how our intravital microscope is used in actual experiments and the overall imaging process from start to finish. We will then finally address any questions you may have during our Q&A session. Now, without further ado, let's begin with the webinar. So as many of you may know, the development of a new drug typically takes between 10 to 15 years and requires an investment cost of approximately 1 to 2 billion USD. Despite this enormous commitment of time and resources, unfortunately, 80 to 90% of drug candidates fail during the clinical stages. In fact, only about 5% of drug development projects ultimately succeed in reaching the market. The drug development process is generally divided into four main steps. First is target identification and lead discovery. Second, preclinical testing. Third, clinical trials. And finally, regulatory approval and post-marketing surveillance. Among all the new drugs that are put into development, around 60% of the drugs that fail when transitioning from the preclinical phase to the clinical phase. And there are many reasons for these failures, but a major factor would lie in the discrepancy between the preclinical and clinical trial results. This is largely due to the limitations of conventional preclinical approaches, such as in vitro assays and endpoint-based methods, which often fail to accurately reflect the drug's behavior and efficiency in living organisms. As a result, the drugs may not effectively reach the intended tissues, or it may not act precisely on the desired targets, or even cause unexpected side effects, all leading to failure in the clinical trials. Ultimately, to address these challenges, we need new technologies that can allow us to precisely observe and understand how drugs behave in real, living biological systems and organisms. So one powerful technology that can help overcome these limitations is intravital microscopy. Intravital microscopy, or IVM, is a high-resolution imaging technique that allows for real-time observation of cellular and tissue-level dynamics within living organisms. Traditional imaging methods, such as in vitro or ex vivo imaging approaches, typically rely on fixed tissue slices or cultured cells. As a result, they lack the ability to capture real-time biological and physiological changes and do not fully reflect the complexity of the in vivo environment. While modalities such as PET or MRI do offer in vivo imaging capabilities, they primarily provide insights at the organ or tissue level and lack the spatial resolution required to observe individual cells in action. And in contrast, intravital microscopy enables the real-time tracking of drug behavior, biological responses, disease progression, and etc. at the single cell level in living animals. It is a powerful tool for visualizing various biological phenomena, including drug delivery and distribution, cell-to-cell interactions, immune responses, and so on, all with high spatial and temporal resolution. And as mentioned before earlier, one of the most traditional methods used to analyze drug responses in the drug development process is the endpoint method. This approach involves sacrificing animals at specific time points to collect tissue samples for histological analysis. However, this method has significant limitations. It cannot adequately capture the variability of drug responses between individual subjects and falls short in providing a precise understanding of drug distribution or mechanisms of action within the organism or animal. As a result, it becomes difficult to accurately evaluate a drug's efficiency, increasing the likelihood of failure in clinical trials. In contrast, intravital microscopy allows for real-time longitudinal imaging and tracking of drug responses within the same individual subject for an over-extended period of time. This makes it possible to precisely determine how effectively the drug reaches the target tissue, as well as the drug's pathway of action, and determine whether the drug causes off-target toxicity in any non-target tissues. Furthermore, because repeated measurements can be taken from the same subject, intravital microscopy can significantly reduce the number of animals required for experimentation, leading to savings in both time and cost. To acquire more precise, reliable, and replicable data during in vivo imaging with intravital microscopy, IVM technology has established an optimized fluorescent labeling protocol specifically designed for in vivo applications. This involves conjugating fluorescent dyes to antibodies and administering them via intravenous injection. Most importantly, the injection dose is precisely controlled at 25 to 33 micrograms, which minimizes variability caused by dosing differences and ensures both accuracy and reproducibility of the same experiment. Moreover, imaging can begin immediately after injection, significantly enhancing experimental efficiency and reducing time overall. This optimized labeling protocol plays a key role in ensuring high-quality data acquisition and maximizing workflow productivity. Now let's take a closer look at the distinct advantages of IVM technology's intravital microscopy and how it stands out from other imaging modalities through various in vivo applications. So to advance research in fields such as immunology and drug delivery, it's essential to understand how and why biological processes occur within the living body at the cellular level. IVM Technology's Intravital Microscope enables dynamic, real-time visualization of cellular responses and disease progression in vivo. This capability provides critical insights that help guide the development of effective therapeutic strategies. Furthermore, by using live animal models, researchers can track the pharmacokinetics and pharmacodynamics of drugs over time or in real time, which is a very powerful advantage in preclinical development. These functions also support the generation of highly reliable data that meet the regulatory standards required for drug development. As shown in the examples here, on the left, we see pulmonary vascular blockage in the lungs induced by inflammation in a sepsis model. And on the right, we can see that the distribution of exosomes and anti-cancer agents, as well as the dynamics and migration of transplanted stem cells, has been imaged and tracked. In addition to its imaging capabilities, IVM Technology's Intravital Microscope is provided as an all-in-one system for easy usage. This all-in-one design system simplifies installation and maintenance, and it eliminates the need for a darkroom, allowing users to focus entirely on imaging and performing experiments without constraints from space or the surrounding environment. The system incorporates several advanced features that ensure exceptional in vivo imaging performance, including a high-speed polygon scanner, real-time temperature monitoring system, and motion compensation technology. We also provide separately an AI image denoiser program, which is an AI-powered noise removal software. The motion compensation feature corrects for organ movement, enabling sharp and accurate imaging, even in organs with significant motion, such as the heart and the lung. The real-time temperature monitoring system allows researchers to track and adjust body temperature during long imaging sessions, ensuring compliance with animal welfare standards and minimizing physiological variation for more reliable data. Lastly, our AI-based noise removal software enables true quantitative analysis by effectively removing noise while preserving the original fluorescent signal emitted from the organ or sample. This ensures clean and precise data sets for further data analysis. Furthermore, IBM Technologies' Intravital Microscope is designed not only for short-term, but also for long-term imaging, ensuring stability throughout extended experiments. To support this, we provide a wide range of customizable imaging windows and chambers that facilitate stable, long-duration imaging across various organs. These chambers also enable real-time pharmacokinetics and pharmacodynamic analysis by allowing direct visualization of drug movement and distribution within the body over time. The current slide here shows examples of some of the most widely used window and chamber designs of IBM technology. If your research focuses on a specific organ or requires specialized conditions, we are happy to assist with the custom development and optimization of imaging windows and chambers tailored to your experimental needs. If you're interested or have any inquiries regarding this, please feel free to reach out to us to discuss further on how we can support your research goals. So by using IBM Technologies' Intravital Microscope, we can visualize various organs in real time in living animals. This enables direct observation of a wide range of biological processes, such as immune responses occurring in vivo, tumor progression and metastasis, drug delivery and mechanism of action, and more. If we take a look at a few examples here, firstly, the drug formulation, we can observe differences in immune activation induced by different drugs. We can also evaluate therapeutic efficacy during cancer metastasis, visualize fibrotic targets for NASH or non-alcoholic steatohepatitis therapeutics, and assess the delivery and efficiency of various drug formulations. These examples illustrate how intravital imaging offers powerful and mechanistic insights across many therapeutic areas, from oncology to immunology and fibrosis. Now I'd like to walk you through a few experimental cases that were conducted by using IBM Technologies' Intravital Microscope. These examples will give you a deeper look into how the system is applied in actual research settings and how it can provide critical insights across different stages of drug development. So the core mechanism of antibody therapeutics lies in their ability to specifically bind to target proteins, thereby blocking pathological signaling pathways or engaging the immune system to eliminate abnormal cells. Therefore, directly observing how effectively an antibody reaches its target tissue, especially in the context of physiological barriers like the blood-brain barrier, is very crucial for understanding its delivery efficiency and mechanism of action. In the following experiments, we visualized the real-time translocation of antibody drugs across the BBB in vivo and quantitatively analyzed the spatial and temporal dynamics of the delivery. Additionally, we examined the distribution patterns of the antibody therapeutics within the mouse brain. So let me begin by briefly introducing the experimental design. We first utilized a Parkinson's disease mouse model implanted with a cranial imaging window. The mice were then divided into two groups, and each group was intravenously injected with a different antibody. Intravital imaging was performed at multiple time points before injection, immediately after injection, and at 24 and 48 hours post-injection. The primary objective of this experiment was to directly visualize and compare the ability of each antibody drug to penetrate the blood-brain barrier. One of the key questions in evaluating the potential of antibody therapeutics for neurological disorders is whether or not the drug can effectively reach its target within the brain tissue. Since the BBB poses a major obstacle by limiting drug access to the brain, tracking the pharmacokinetics and distribution of the antibody drugs in real time is essential. This experiment provides a platform for both quantitative and qualitative analysis of BBB permeability, serving as a critical preclinical step in validating the feasibility of antibody-based therapies for brain tumors and central nervous system disorders. So here are the results from the experiment. The antibody drugs were labeled with green fluorescence, while the blood vessels were visualized with red fluorescence. Although there were some variations between the animals, antibody fluorescence signals were observed in the brain tissue at 24 and 48 hours post-injection for both antibody drugs. Overall, antibody drug accumulation was consistently observed within the vasculature, notably based on the regions marked with the asterisks, which indicate presumed sites of antibody drug penetration. Antibody drug 2 appeared to exhibit greater penetration into the brain parenchyma compared to antibody 1. These findings were further supported by quantitative analysis, as shown in these graphs here. In graph A, we observed that the fluorescent intensity of the antibody drugs in the brain increased at both 24 and 48 hours after injection, indicating successful delivery and retention. Graph B here illustrates the differences in kinetic profiles between the two antibody drugs. The gray graph here represents the antibody drug 1, and it shows a gradual increase in fluorescence signal over the 48 hours, with some inter-animal variability. In contrast, the red graph represents antibody drug number 2, and it exhibits a rapid increase in signal at the 24 hours post-injection point, followed by a stable and sustained intensity thereafter. And in the same experiment, the distribution patterns of each antibody drug within the meninges were also observed. Although some inter-animal variability was present, overall, the antibody drug number 2 exhibited a higher level of uptake compared to antibody drug number 1. These results suggest that the accessibility and accumulation characteristics of antibody drugs within the meninges may serve as important factors influencing delivery efficiency and therapeutic target selection. And in the next following experiment, two groups of mice were each injected with a different antibody drug, and the distribution patterns were observed in real time to compare the uptake of the two antibody drugs within the brain. So first, a cranial imaging window was implanted into the mice, which was followed by a 2-week recovery period. Then the mice were divided again into two groups, and each group was intravenously injected with a different antibody drug. Imaging was then performed repeatedly at different time points, which were before the drug was injected and immediately after the drug was injected, then 6 hours, 24 hours, and 48 hours post-injection. This experimental design aimed to observe how effectively the antibody drugs cross the blood-brain barrier and to monitor changes in drug distribution over time. Additionally, the study sought to compare which of the two antibody drugs exhibits higher BBB penetration efficiency. The experimental results are here as follows. First, the drugs were labeled with red fluorescence, and the blood vessels were labeled with green fluorescence for observation. It was confirmed that both antibody drugs were delivered to the brain tissue via the blood vessels starting from the 24 hours post-injection time point. In particular, compared to the single antibody drug, the double antibody drug showed a wider distribution range within the brain. Furthermore, a similar trend was maintained at the 48-hour time point, with the double antibody drug exhibiting a higher level of uptake and a more extensive distribution pattern within the brain tissue compared to the single antibody drug. And if we take a closer look, as shown, the antibody drugs labeled with red fluorescence were mostly confined within the blood vessels up to the 6 hours post-injection time point. However, from the 24-hour time point and onward, migration into the extravascular tissue, specifically the brain parenchyma, was observed, which was a common pattern for both drugs. Notably, at the 48-hour time point, the double antibody drug exhibited a wider and greater distribution pattern within the brain tissue compared to the single antibody drug. This suggests that the double antibody drug had a relatively higher delivery efficiency into the brain tissue. A similar trend was also observed at other spots as well in the brain. In this case, drug uptake into the brain tissue began from 24 hours after antibody drug injection, and at 48 hours, the double antibody drug was found to be more abundantly distributed in the brain tissue. So as before, antibody drug uptake into the brain tissue was confirmed to begin from 24 hours after injection, and at 48 hours post-injection, it was observed that the double antibody drug accumulated at a higher level within the brain tissue compared to the single antibody drug. This suggests that the double antibody drug has higher brain delivery efficiency. These results provide direct visual evidence that the antibody drugs actually reach their target sites within the brain. By imaging the detailed tissue distribution and delivery routes, information that cannot be determined from blood concentration alone, we can more precisely evaluate the efficiency and limitations of drug delivery strategies. This approach can serve as a critical validation tool in preclinical stages for demonstrating the therapeutic potential of antibody drugs developed for treating brain diseases. And again, as mentioned earlier, antibody therapeutics are widely used in various cancer treatments due to the high specificity for targets and strong therapeutic effects. However, how uniformly these drugs penetrate and act within the tumor and through which pathways they distribute remain unclear. In particular, tumor heterogeneity and the microenvironment of solid tumors can act as major obstacles to drug delivery, which may reduce treatment efficiency and lead to drug resistance. Therefore, to precisely predict and optimize the therapeutic efficiency of antibody drugs, it is essential to have technologies that allow direct in vivo observation of actual drug distribution and mechanisms of action within tumors. Next, addressing this need, I will introduce experimental data that visualizes the intratumoral distribution of bispecific antibody drugs using intravital microscopy. So to enhance, as mentioned earlier, to enhance the efficiency of antibody therapeutics, it is crucial to precisely understand how the drug distributes within the tumor and how efficiently it reaches their target cells. In particular, bispecific antibody drugs have the advantage of simultaneously recognizing two distinct antigens, enabling them to target both tumor cells and immune cells to induce or amplify immune responses. However, it's difficult to accurately understand how these bispecific antibodies distribute and act within the tumor microenvironment without real-time in vivo observation. Therefore, directly visualizing the distribution and penetration of drugs within the tumor tissue in vivo plays a key role in elucidating mechanism of action and evaluating therapeutic efficiency. So based on this need, the present study analyzed the intratumoral distribution patterns of bispecific antibody drugs through real-time in vivo imaging. So for this experiment, we first established a tumor model by implanting a dorsal skin fold chamber in the mice, and then we injected cancer cells to create the tumor model. And then the mouse was injected with the bispecific antibody drug and also with antibodies to visualize the blood vessels in the mice with red fluorescent signals. And then imaging was performed at 4 hours and 24 hours post-injection. This approach allowed direct observation of how the bispecific antibody distributed within the tumor environment and how its pattern changed over time. So here are the observed results. At the 4-hour post-injection, the bispecific antibody drug labeled with the green fluorescence was mainly confined within and around the blood vessels. In contrast, after 24 hours, the antibody drug gradually penetrated beyond the vasculature and deep into the tumor tissue, showing clear co-localization with cancer cells. This time-dependent change in distribution suggests that the bispecific antibody drug not only travels through the bloodstream, but also effectively diffuses and acts within the tumor microenvironment. The 3D image analysis further clarified these distribution patterns. At 24 hours post-injection, compared to the 4-hour time point, the drug was observed to be more localized within the tumor tissue and showed stronger binding affinity to the cancer cells. This suggests that the bispecific antibody drug has a high binding affinity for the target cancer cells. Additionally, with prolonged imaging, it is expected that the actual process of cancer cell destruction may be observed in the future. These experimental results provide visual confirmation that the drug possesses delivery characteristics that precisely target tumor tissue, supporting the fact that the bispecific antibody drug actively migrates and acts within the tumor over time. This real-time in vivo imaging technique demonstrates itself as a highly useful tool for quantitatively and spatially evaluating the pharmacokinetics and target specificity of antibody therapeutics. So far, I've introduced application data using IV Technologies Intravital Microscopy. Lastly, I will show you the imaging process using IV Technologies Intravital Microscope through a brief demonstration. So in this demonstration, we visualize the vasculature and neuronal structures of the mouse brain. And by using a transgenic mouse, the neurons in the brain were labeled with a green fluorescent signal, and the blood vessels were labeled with a red fluorescent signal. We then used a 2-photon laser microscope to observe the fibers within the brain. So what you are seeing now is the engine screen of IV Technologies Intravital Microscope. Before starting imaging, you can adjust the laser power and detector gain values located below. And after setting these values to your desired levels, you can use the jog dial to move the stage to find your desired position and proceed with imaging according to the selected imaging mode. And after imaging, the acquired data can be further edited using the studio software such as inserting scale bars, adjusting the brightness and contrast, and performing additional processes like mosaic stitching and Z projection. The screen you are currently viewing here shows mosaic stitching process data. And in the 3D mode, you can view the imaging data's depth at a glance, and through 3D recording functions, you can perform not only 2D, but also 3D analysis with your acquired data. So these are the imaging data of the blood vessels and neuronal structures of the mouse brain obtained using the method demonstrated earlier. On the left is the result from confocal imaging, and on the right is from the two-photon laser imaging. You can see that the neurons labeled with the blue fluorescent signal are densely distributed around the blood vessels, and the fibers that were difficult to observe with confocal microscopy were clearly visualized using the two-photon laser, where they were labeled with a blue fluorescent signal. And this is the 3D analysis video of the same results. In the video on the right, you can view the brain from the surface down and see the entire depth of the brain all at once. And in the video on the left is an orthogonal view of the brain in which you can observe each layer starting from the surface, allowing a detailed examination of how the blood vessels, neurons, and fiber structures are arranged in each layer of the brain. In addition to the dorsal skin and brain imaging demonstrated earlier, high-resolution intravital imaging is also possible in various organs of living mice, such as the lungs, the spleen, and the heart. Moreover, IVIN technology not only provides an all-in-one system capable of high-quality intravital imaging, but also offers CRO services based on researchers' needs. Through this, we can support more efficient and professional research execution. So what you are seeing here now are actual customer cases where we conducted CRO services together. We collaborate with various companies, research institutes, and universities to provide CRO services for both short-term and long-term imaging projects. I'll now briefly introduce the workflow of how our CRO service is carried out. So the CRO service provided by IVIN Technology follows the process outlined below. First, we conduct a detailed discussion of the experimental scheme through consultation. Next, a preliminary test called the feasibility test is performed to verify whether or not the proposed experimental scheme can be properly executed. Once this stage is passed, we proceed to the main experiment phase, where in vivo imaging is conducted and data is collected. Finally, after post-processing and analysis, the experimental results are compiled into a final report and sent to our clients. For more detailed inquiries about our CRO service, please contact us at the email address below, or you can visit our website at www.ivintech.com. So thank you everybody for joining us for today's webinar. Over the course of the session, I explained why intravital microscopy is vital to the overall drug development process and how it can be applied to drug development. I also highlighted the advantages of IVIN Technology's unique intravital microscopy system and shared some application cases acquired with our system. We sincerely appreciate your time and attention, and that concludes my presentation. Thank you.
Thank you for the detailed presentation, Jenny. So now we'll move on to answer some questions. So if you have any question, please use the Q&A box below. So, okay. So the first question is, how long does the fluorescence signal last when you label the blood vessels?
So the fluorescent signals typically last up to 24 hours. And if you would like to image beyond 24 hours, we recommend injecting an extra booster shot. So from the day 2 point, you would inject about 25 micrograms more of the antibodies conjugated with the fluorescent dyes to really maintain that fluorescent signal throughout your experiment.
Okay, then the second one is it possible to tag other molecules with the fluorophore using I-beam technologies protocol, not just antibodies?
Yes, so we mainly do conjugate antibodies, but other than antibodies, we're able to conjugate our dyes with different samples of coming from researchers. For example, we're able to conjugate it to protein drugs or other different molecules that can be used in your experiments. If you maybe would like to ask or inquire if your sample is able to be conjugated with our dyes using our protocol, please give us an email at the email address you can see on the screen.
Okay, I think it'll be the last question for today's session. So the last one is, once you implant the chamber, how long can it be kept in place?
So for the chambers, it can be different according to the organ, but if I give you a brief explanation using our cranial imaging window, the mouse can survive up to shortly from 6 months and maximum a year after having gone a very good recovery period and after having really inhibited the inflammation that can be caused by the surgery. And another example would be the liver imaging window, which also after taking care of inflammation after surgery, it can last from a couple of weeks to maybe a month or two.
Okay, thank you. And Jenny, there's one more question. So is it a confocal or two-photon system? What are the working distances?
So our system has both a confocal and a two-photon laser incorporated into the system. The main difference would be for the two-photon laser, we have a fixed laser system, which is fixed at a 920 nanometers. And we have another system where the two-photon is a tunable laser, so you can change the wavelength of the two-photon laser according to your research experiment conditions. So it is not separately a different model. The model 1 has both a confocal and a two-photon laser system put into the system, and the working distance would be different according to the objective lens that you would be using. If you would like any more technical details about the technical specifics behind our system, you can again give us an email and we can answer your questions through the email for you.
Okay, thank you for answering the questions, Jenny. Thank you everyone for your questions. So the webinar recording will soon be available on our YouTube channel. At I-beam Technology, we offer both hands-on demos at your site, starting from a couple of days to weeks. And we also offer live virtual demos through Zoom. We can also test your samples and provide training for new animal surgery model for long-term or terminal intravital imaging. If you have any more questions or if we couldn't address your inquiry during the Q&A session, please feel free to reach out to us at information@ibeamtech.com. We appreciate your interest in I-beam technology, and we thank you again for joining us today. We look forward to seeing you again. Thank you.